Hi, and welcome to this run-through video for the 2018 Breadth in Biology paper. So there's two ways you can do this. Either you can have the paper printed out, if you follow the link below, and kind of go through it with me, or you can do this and then you can use this video to mark it. So let's get started. The first thing I'm going to do is I'm going to show you the mark scheme for the multiple choice section of the paper, 20 questions. And that way you can quickly mark it and then you can just watch the parts of the video if you're not sure where you went wrong. Okay, so first of all, here is the mark scheme for the multiple choice section. Pause the video and use this to mark your paper. Okay, so now let's go through the multiple choice so we can sort of work out how you get to those answers for your understanding. Let's go. All right, number one. Which organelle is not involved in the production and secretion of enzymes in eukaryotes? So the best way to do this is always just kind of like go through it. So that is, yep, secretion, ribosomes, that is. This isn't, okay? This is. This one, smooth is the key word, because smooth endoplasmic reticulum is about lipid and steroid synthesis, whereas the rough endoplasmic reticulum is involved in protein synthesis. So the answer is C. 2. Autoimmune diseases are often treated with a course of antibody injections. Which of the following statements describes the immunity arising from this treatment? Okay, so this would be antibody injections. So this means that it is a passive artificial immunity. So what this means, passive is injection of antibodies as opposed to allowing your body to create its own antibodies, which would be active. And it's artificial because it's an injection. So quickly, active, natural, that's basically you getting a disease once and then becoming immune to it. Active, artificial, that's being vaccinated. Passive, natural, that is basically babies get antibodies from breast milk and they become naturally passively immune because they're not making the antibodies, but the mother is. On to 3. A student designed an investigation into the rate of transpiration in plants. They used 8 leaves of the same size, age, and species. They kept environmental conditions such as wind speed, temperature, and humidity constant. Okay, so this is a question about sort of how science works. To make the investigation valid, to increase the accuracy of their readings, to increase the repeatability of their data, to improve the precision of their results. I would say repeatability is the best one, because it's about having it. I mean, you could almost say it's accuracy because it's you would probably have a more accurate average result. But yeah, with more data, you're going to get a more repeatable result with 8 leaves kind of averaged. So I think C is the best one. But that's a bit of a tricky one. 4. The image below shows isomaltulose, a disaccharide from alpha glucose and fructose. Right, okay, I must confess, I just had to edit this video because I got this wrong first time round. The answer is A, alpha 1,6 glycosidic bond, and this has reminded me that I need to be better at labeling the carbons in the fructose molecule. So this is fructose here, and this is the alpha glucose here. Okay, we should probably be quite good at labeling the carbons in glucose. So it goes 1, 2, 3, 4, 5, 6. Now fructose is a bit harder because we have to start down here. Okay, so this is 1. Then that is 2. So it goes 1, 2. Then this is 3. This is 4. This is 5. And up there is 6. So which one is it? It's an alpha 1,6 because it's between carbon 1 of the glucose and carbon 6 of the fructose. Okay, that's a tricky one. I got that wrong first time round. I had to check the mark scheme, but that's how you get there. 5. The graph shows the rate of movement of 4 different substances across a membrane. The substances shown in the graph are carbon dioxide, testosterone, which is a lipid-based hormone, that's key, ethanol, and sodium ions. Okay. Which of the patterns A to D represents the pattern of movement of sodium ions across a membrane? And the answer is going to be D, because sodium ions basically do not travel across a membrane unless there is a channel protein, okay, or transport protein. So when the sodium ion concentration goes up, eventually the channel proteins are going to become saturated. So it's kind of a bit like a rate of reaction graph for an enzyme. When you increase the substrate concentration, basically they get, the channels are working at their maximum rate, and that sort of the number of channels becomes the sort of limiting factor. So the answer is therefore D. Yeah. Okay, 6. The aquatic crustacean Daphnia magna has a heart that pumps a blood-like liquid called hemolymph around the body cavity. Okay, so this is an open system. It's probably single. Yeah, I don't think there is any double. No, there's no double open, I don't think. So it's a single open one. So open circulatory systems don't have blood vessels. They just have the heart, which kind of sort of pushes hemolymph generally around the body. And I don't believe I know of any open ones which have a double system. Yeah, that couldn't be true. So yeah, single open. By the way, just in case you're wondering, we have a double open. So sorry, we have a double closed. That's us. Double open, I don't think exists. Single closed would be something like a fish. Okay, on to 7. DNA polymerase. Whoops. DNA polymerase catalyzes the formation of phosphodiester bonds during DNA replication. Which statement A to D will not affect the rate of phosphodiester bond formation? Well, A certainly will. The length? No, I don't think that will. pH certainly will. This is basically the substrate concentration. So yeah, B is the one. Because we're talking about the rate of bond formation, not the time to finish the whole thing. So yeah, 7B. Which of the following types of cell is not involved in a primary immune response? T memory. Yeah, anything memory. Yeah, because the memory cells only come into play on your secondary immune response, or the second time you've been infected. The memory cells mean that you can sort of fight your infection a lot faster. T memory, remember, we also have we have T memory, and we also have B memory cells that lead to immunity. 9. Which of the following A to D is not an adaptation to reduce water loss in plants? To reduce water loss, I think that's the key thing, okay? To reduce water loss. So don't get tricked into thinking, oh, well, this is good for water, it's going to help the plant. No, it's about the loss of water. So this, I think, is A. I think straight away, because A is not about water loss, it's about getting extra water. This would reduce water loss. This would reduce water loss because the stomata only open at night. And this would also reduce water loss. Yeah, so it's A. All of those would be potential adaptations of a xerophytic plant, like this one. But only A is not to do with water loss. 10. A scientist was investigating the effect of 2 different temperatures on the rate of enzyme-controlled decomposition of ammonia in soil bacteria. They repeated their experiment 10 times for each of the 2 different temperatures. Which of the following, A to D, statistics, should they use to determine if there was a significant difference between these 2 sets of times? Well, a significant difference is normally t-test. Yeah, because you can pair, yeah, it's t-test, I think. All right, 10? Yeah, t-test. So standard deviation is just for looking at the spread of data for one mean. Students' t-test is really about comparing 2 means. 2 means, and you have to have, the mean, obviously, for each data set, and you have to have the standard deviation for each data set. And then what you're basically doing is you're kind of looking, if that's one standard deviation there, whoops, this is like along an axis, and then we have a sort of another standard deviation, distribution there. What we're doing is really trying to sort of see, are those 2 distributions, do they overlap enough that we could sort of say that they're basically the same, or are they separate enough that we can say, yeah, this is 2 different things going on. 11. The hydroxyl OH group of carbohydrates is polar and makes the molecule soluble in water. The greater number of free hydroxyl groups as a proportion of the number of carbon atoms, the more soluble the carbohydrate. Ooh, okay. Okay. All right. Glucose is going to be the most soluble, I think. Okay, so definitely, yeah, it's going to be, what could be ribose? So okay, so how am I trying to work this out? I haven't looked at the answer yet, so I don't know what the question, the mark scheme is going to say. But so a carbohydrate is CnH2O, well CH2O, sorry, all to the N, pretty much. Okay, that's a carbohydrate. So whether it is 5 carbon or a 6 carbon sugar, I don't think should matter. So I think it's going to be, okay, well, it's either going to be A or B, because this A here is glucose ribose. So monosaccharides have got the most free OHs. So these are 2 monosaccharides, these are 2 monosaccharides. The reason not this one, even though that's a monosaccharide there, is that glucose is down here, which I think, no, that can't be right. So we've moved to A or C, so we can cross out B, and then we're just looking to see which is the most sensible order. Now, amylopectin is going to have more free N's than amylose. So I think amylopectin is going to have more free OHs than amylose is. I think. So I'm going to say it's C, would be more soluble. Is it? Oh, no, it's A. They've said amylose. So they've said amylose is more soluble than amylopectin. That's weird. Okay, so the answer is A. Now, let me try and understand why that is. Edit. Okay, so I've had to think about this question. I think this is why they've said it, although I'm not entirely sure if I really think this is a brilliant question. I'm not really sure if I agree with them. I have to do some more research, to be honest. But this is the structure of amylose, okay? It's a spiral. It's a straight chain, but it's kind of coiled on itself. So if we think about the free OHs, there's free OHs all the way along the chain, okay? So there's lots of free OHs. It's only the only the OH on carbon 1 plus carbon 4 are sort of used in bonding. Now here, this is amylopectin. So amylopectin is basically the same idea, but you've got these branches coming off, which I'll sort of highlight in different colours. So here's a branch, here's a branch, here's a branch, here's a branch. Now if you think about that, which carbons are involved in the bonding? So we've got carbon 1, yes, we've got carbon 4. We've also got carbon 6. Okay, carbon 6 is also involved in bonding. So that's this one here, and this one here. So, okay, so this is why they say that amylopectin is less soluble than amylose. But I actually think this is wrong, OCR, if you're listening. I think this is wrong because you have forgotten, OCR, I believe, to consider the fact that for the same number, if this was the same number of carbons in the molecule, then that would be sort of, these ones would be sort of added in the chain. So you've kind of forgotten to consider the effect of the extra end points, I think. So I actually think OCR is wrong on this one, but they've said A, so we'll go with that, okay? But get back at me, OCR, if you see this video. All right, moving on to 12. Okay, the bacterium Parenchyma cellulosum and the fungus Armillaria are both found in the soil. Which of the following shows the structures present in each organism? Okay, basically we've got bacteria, and we've got a fungus. So we're just comparing those two. All right, so let's... Free ribosomes in the cytoplasm. Yes, definitely it's both, okay? Everything has a ribosome, so we can immediately cross off C and D. Great, we have to read less. Membrane-bound nucleus. Yeah, it's only the fungus, so that's good. Okay, well, doesn't give us anything. DNA in a single loop. Well, that's only the bacteria, so it's not this one. Let's double check this. Cell wall present. Well, it could be both, in fact. Probably is both. So, yeah, it's A. That's easy. All right. 13. Carbon dioxide released during respiration can affect the percentage oxygen saturation of hemoglobin. Yes, it can. Tertiary structure of hemoglobin is affected when carbon dioxide reacts with water to form carbonic acid. This reaction releases hydrogen ions. Which of the statements A to D explains this change? Okay, well, this is where we just find mistakes. The release of hydrogen ions causes the pH to rise. No, it doesn't. It causes it to fall. So that's wrong. The release of hydrogen ions causes the pH to rise. Again, that's wrong. The release of hydrogen ions causes the pH to fall. Correct. Which increases hemoglobin's affinity for oxygen. No. So when we have hydrogen ions, it decreases the affinity. So this is wrong. So this last one's going to be right. pH to fall, which reduces the affinity. Yes. So the answer is D. This whole thing is called the Bohr effect, or the Bohr shift. This kind of principle. All right, moving on. 14. During translocation of photosynthetic products in the phloem sieve tube, hydrogen ions are moved out of companion cells and they move into the sieve tube elements. Then sucrose enters the companion cells and moves through. Sucrose enters the companion cells and moves through plasmodesma into the sieve tube. Oops, sorry, I misspoke there. It's the H+ ions go out of companion cells and then it comes back in, sort of with co-transport of sucrose. So enter or leave the companion cells. Okay, so hydrogen ion out of the companion cell. This is active transport. So it's either C or D. Sucrose into companion cell. We would sucrose into companion cell, facilitated diffusion. Yeah, it's actually sort of co-transport. And sucrose out of companion cell is diffusion. So yeah, it's 14C. Just draw a quick picture of that here. Draw it in green. So we've got, this is the sieve tube element here, kind of, sieve plates and so on. And then here we've got the companion cell. Right, CC for companion cell. Now actually there are these tiny little, it's going to rub out the whole thing, isn't it? With these tiny little plasmodesmata which link the companion cell to the sieve tube element. So what happens is H+ actively transported out, so active transport, and then the H+ can passively come back, but it has to be with sucrose. So this is actually an example of co-transport, which is a sort of subcategory of facilitated diffusion. So the sucrose can only come in if it comes in with an H+. So we use the H+ gradient to bring the sucrose in. And then when the sucrose molecules, which I'll draw like this, are kind of in there, they come in at sort of high concentration, and then they can just passively diffuse through the plasmodesmata into the sieve tube element, okay? And that's what powers translocation with the phloem. All right, moving on, 15. Millennium Seed Bank has over 2 billion seeds in storage. Which of the options A to D describes the type of conservation by the Millennium Seed Bank? Okay, well, first of all, we have to look, is it in situ or ex situ? It is not in situ, because that's like conserving a rainforest where it is, in the situation, in the place where it is. So it's definitely not in situ. It is ex, so it could be, this is right. But is it conserving species or habitats? Well, it's conserving species, because seed bank can't preserve a whole habitat. It's just preserving the species that might go into that habitat. So the answer is 15C. 16. Plants such as the soybean have a number of defense strategies to prevent infection by pathogens. Which of the following strategies is a chemical defense against pathogen infections? Oh, okay, chemical. All right, so callos, you might have been sort of tempted by this one, but this is actually a physical block. Okay, so Callose is essentially a plug that plugs the sieve tube. Let's just go back up here, actually. So sometimes if a virus, let's say a virus gets into the sieve tube elements, maybe because an aphid comes and punctures the bark of the tree and injects a virus with its saliva in there, then what happens is basically Callose, which I'm just going to draw as blue gunk, blocks the sieve tube element. So the virus is kind of stuck in this block and it can't move through the phloem. So it's a physical block. All right. This one is probably going to be a chemical defense because it's a hydrolytic enzyme. It's a chemical defense. This again is physical, just physically closing stomata. And this again is physical. So the answer is 16B, the difference between physical and chemical defenses that plants have against pathogens. Oops, gone to the top again. Moving on down to 17. Swiss chard is a leafy green vegetable related to spinach. It's quite tasty. Some varieties have yellow stalks that have vacuoles containing yellow beta-xanthin pigments. The graph shows the effect of temperature on the release of these pigments. Okay, so you may have done this Beetroot Practical in class in year 12, where you sort of heat up the beetroot and you see kind of how much that red colour leaks out. The exact same idea, except with just a different pigment, which happens to be yellow. So, it was deduced that the beta-xanthins were released from the vacuole due to the denaturing of proteins in the tonoplast vacuolar membrane. I don't like this question really. Which temperature do they denature? Well, it's around C. Because 17C, yeah. Because at this point, the membrane breaks down. Okay, the reason I don't like this, OCR, for listening, is because is it really about the denaturing of proteins in the tonoplast? I mean, maybe it is. I don't think it is. I think it's just that the phospholipid bilayer loses its integrity at a certain temperature. I don't believe that the proteins in the membrane are needed for the structural integrity of the membrane. I think it's just the fact that the phospholipid bilayer is jiggling about so much that it kind of breaks down. So that's my view. 18. An investigation into how a change in sodium chloride concentration affects osmosis in potato cells concluded that the isotonic point of the potato was 0.25 molar. Investigation into how a change in sodium chloride concentration affects osmosis. Okay, so this is basically saying that the concentration of sodium chloride in the potato was 0.25. And so the isotonic point is when the concentration of sodium chloride inside the potato cells is the same as the concentration outside the potato cells. And in which case there would be no net movement of water between the cells and the external solution. So there is a net movement of water? No. There is a net movement of water? No. There is no movement of water. Oh, I almost got tricked here. I almost went for this, but actually, yeah, there is movement of water. It's just the movement of water into the potato cells is equal to. So if this is a potato cell here, let's just picture one of them, and this is the solution outside. There is movement. Water molecules are crossing this way, but they're also crossing this way. So the movement is equal. We could say there's no net movement, but I wouldn't say there is no movement because individual water molecules are still going backwards and forwards just at the same time. Okay, so that is going to be D. All right, moving on. 19. Biological molecules and the component elements. Okay, so sucrose is a sugar. So yeah, it's not this one because there's no nitrogen in sucrose. Carbohydrate, carbon, hydrogen, oxygen. Now cholesterol is a lipid, so it doesn't have nitrogen in either. It's still carbon, hydrogen, oxygen. But remember, lipids have less oxygen, but there is still oxygen there, it's just less of it. Insulin is a protein, is what you've got to remember here. So proteins do have nitrogen and sulfur because sulfur is in the disulfide bonds, which are often present in in proteins, it's found in the amino acid cysteine. So it's going to be one of these, not this one, phosphorus. So we've crossed out all the ones that can be. It's got to be C, but let's just hope this is correct. So ATP, yes, it does have phosphorus. That's the phosphate ATP. It does have nitrogen because adenosine A is a base. Okay, adenosine triphosphate. It's basically a nucleotide with some phosphates added. So the answer is C. 20. In human cells, the tumor suppressor gene, P53, codes for a protein that interrupts the cell cycle if there is any damage to the DNA and prevents the copying of damaged DNA. In which of the stages could TP3 interrupt the cell cycle? Okay, so prevents the copying. This is the key phrase. Okay, so the P53 has to act before the DNA is copied. So it has to be before S phase. It's not in S phase, it's before S phase. So now we need to kind of just look at the cell cycle. So mitosis is the start of it, isn't it? And then we have G1. And then we have S phase. And we have G2. Oh, and cytokinesis is there. So where is it going to do? It has to be before S, so therefore it has to be in the G1 phase. And actually, if you really want to get technical, it works sort of here. There's a sort of G1S checkpoint before we go into S phase, and that's where the P53 is working. Right, okay, so that's the end of the multiple choice section. Pause if you want, and now we're going to get straight into the more longer asked questions, okay? So here we go. Okay, here we are, question 21. Now this is all about a water frog that lives in Lake Titicaca. So when I was reading this, preparing this video, I thought I'll put in some pictures of me on Lake Titicaca because I went there about 3 years ago. So here's me sitting on Lake Titicaca over here on the right, looking out across this beautiful reflection and sunset. And the thing about Lake Titicaca is it's the highest, I think it's one of the highest altitude lakes in the world. So it's actually at 3,800 meters altitude, and that information is sort of helpful for this question. They didn't really explain where this lake is, but the lake is at very high altitude in the Andes Mountains. Anyway, let's get into the question now. So here's the frog. It is called Temlatobius quelus. It's an aquatic amphibian found in Lake Titicaca. Now, can you remember sort of this mnemonic? Did King Philip come over from Great Spain? Domain, kingdom, phylum, class, order, family, genus, species. And it's the last two that form the name, the Latin name of or the scientific name of the frog. So the genus is this word here, Temlatobius. And you've got to copy it correctly to get the marks, so make sure you do that. Oh, I've just messed it up, didn't I? Telmatobius. Telmatobius. All right, outline the properties of water which make it an ideal habitat for an amphibian. Well, I think the first thing, kind of linking it to the fact that this is a high altitude lake, is it has a high specific heat capacity. And the second thing is, which means that it's thermally stable, high specific heat capacity, which means it's thermally stable, or it doesn't change temperature so much. And the other thing that I always like to remember is that ice is less dense than water, which means that it will, if it does freeze, the ice will float to the surface and it actually acts to insulate the lake. And this is really important because it does freeze up there at 3,800 meters from time to time, I'd imagine, and that helps the frogs survive. So let's quickly check the mark scheme. And here it is. Okay, so Telmatobius, notice here that you've got to have a capital T. If you don't have a capital T, you don't get the mark. And this is the thing that I said, ice is less dense than water, and we also talked about high specific heat capacity or stability. You could have also had, it's a good solvent, it has oxygen dissolved in it, and yeah, it has a high density, so the frog is floats, I guess. Oh, didn't know that. Okay, right, moving on. Back to the paper. All right, so this frog, it looks pretty weird, doesn't it? it's got sort of these folds of skin, these large flaps of skin. It says in the question, suggest why the Titicaca water frog has involved unusually large folds of skin. Well, it basically gives it a larger surface area. To absorb oxygen. And the reason why it's helpful to know that this is a high altitude lake is because there's a low oxygen concentration in the water, because there is less oxygen the higher up you go, and at 3,800 meters, more or less, altitude, there's not that much oxygen. It's quite difficult to breathe up there coming from sea level. So the mark scheme sort of says this sort of stuff. So reference the increased surface area, or you could say the surface area to volume ratio, idea of increase in oxygen absorption, and because the oxygen levels in the lake are low. So you could get these two marks without any knowledge about Lake Titicaca. This one, it would have helped to know that it is this unique kind of environment. Moving on. When out of the water, the Titicaca water frog is able to use its lungs to absorb oxygen. Lungs contain specialized gas exchange surfaces. Describe and explain how one feature of the lungs provides an efficient gas exchange surface. Describe, okay, well this is interesting here because it's a little exam technique. We don't want to just list everything we know about the lungs. We want to describe and explain how one feature, so like say it, explain it. So for example, large surface area of the lungs gives a high, large surface area of the lungs gives a large space for the absorption of oxygen so that oxygen can be absorbed rapidly. Or you could say the lungs have a very thin membrane of the alveoli, or very thin epithelial layer of the alveoli, which means that oxygen can move rapidly from the air to the lungs. Or we could say that the lungs have a very good blood supply, which maintains a high concentration gradient. So you have to pick one of those things and explain. The mark scheme kind of shows that. So each of these pairs on the mark scheme here, D and E, is a describe and an explain. A describe and an explain. And a describe and an explain. So you can't get, let's say, this mark here for the describe and then explaining this mark. You know, you have to get both marks of the pair there. Okay, so pause that if you need to have a little look, and we're going to move on. Okay, a student was investigating the effect of cell size on the rate of diffusion into model cells. Rate of diffusion into model cells. The student was investigating the rate of diffusion into model cells. They had 2 cubes of agar containing phenolphthalein indicator. The student placed the cubes in dilute acid, which caused the indicator to become colourless. They measured how much each cube became colourless over time. Suggest 2 ways they could have ensured they had confidence in results. Okay, so I think this is talking about how to have confidence in, this is an interesting phrase, how do you have confidence in their results? How are you sure it's not a one-off is what I read that as. So I think we need to do this multiple times. Multiple times with similar clues. And then take an average. After eliminating anomalies, maybe. But this is an interesting way. This is an interesting way of language about this question. I haven't seen this kind of language before. How would you ensure they have confidence in their results? Let's see if I'm right on the mark scheme. Yeah, okay, so that's what it's going for. Repeat findings, calculate mean, identify anomalies. There's another one which I didn't think of. Use a statistical test to identify the differences. And maybe you could do the t-test, for example, because that's about investigating the difference between 2 calculated means and seeing if there is a statistical difference between those 2 means. Okay, moving on. Okay, cube A is 10 millimeters long, and cube B is 4 millimeters long. Calculate the surface area for both Cubes. Okay, well, first thing, if you have forgotten how to do this, you need to think about what is a cube as a net. If you're familiar with the concept of a net in mathematics, the net for a cube kind of looks like this. If you fold it up, so therefore there are 6 faces to a cube. Okay, that's going to help. All right, so the surface area. Let's just do, so which one? A is the big one, and B is the small one. All right, so surface area for A is 6 times 10 by 10, and for B is 6 times 4 by 4. So that is going to equal 600 here, and 4 by 4 is 24. No, so what am I talking about? 4 by 4 is 16. Times 6 equals 96. Okay, so the surface area is 600 and it's 96. What about the volume? The volume for A is 10 by 10 by 10, or 10 cubed, which is equal to 1,000. And the volume for B is equal to 4 cubed. So it's 4 times 4 times 4, which is equal to 64. So the surface area to volume ratio, so remember it's the SA divided by the V. So therefore, we've got 600 divided by 1,000 for A. 600, put a little divided by 1,000, which is equal to 0.6. And for B, it's 96 divided by 64, which is equal to 1.5. Okay, tick. In terms of when you get the marks for that, like if you didn't get all the way, can you get some of the marks? Probably. Oh, it's 1 mark for each, basically. 1 mark for the 0.6 and 1 mark for the 1.5. You actually will get the marks. You can get 1 mark for this, for not sort of simplifying the ratio and giving it as a standard decimal to 1, but you could get a mark for this over here. Moving on. Explain why the surface area to volume ratio of an organism determines whether it needs a circulatory system. Well, we need to sort of talk about if SA to V is big enough, then there might be no need for a specialized exchange surface, such as the lungs or the gills of a fish. And so you could say enough O2 absorbed, for example, and also you could maybe say if the organism is very flat, then all the cells in its body might be close enough to the external surface that they can get oxygen without having it pumped around the body. So I'm thinking of a flat worm, which is basically like a, well, pretty flat worm. It doesn't have eyes, but I like to draw it with eyes just because it makes me think of what it's like. And this is a flat worm like that. Let's look at the mark scheme. Okay, so I approached it from this angle. ORA in a mark scheme means or reversed argument. So I said if the organism has a big enough surface area to volume ratio, then uptake of oxygen might be sufficient. But you could have said the opposite. So if the organism has a small surface area to volume ratio, then diffusion is too slow and the diffusion distance is too long for sufficient delivery of oxygen or removal of waste products for aerobic respiration or metabolic demands. I think I'm not sure if I would have got 3 marks there. I think I might have got 2. I think this is a key word here. Always linking it back to keywords. Respiration, I think, was something I didn't say. So why do we need oxygen? I forgot to say the obvious. Sometimes that's what I forget to say. So needed for respiration, keyword. Don't forget to make the obvious points. Okay, moving on. All right, here's a triglyceride molecule found in sunflower oil. Circle an ester bond. I'm going to highlight it actually, okay? I'm going to highlight it in blue. So an ester bond is this. Okay, now you have to highlight that whole part there. So you've got to highlight both oxygens there, and also this central carbon. Okay, so there's one. I'll just highlight the other two as well. So any of these would be fine, but you must circle both oxygens and the carbon. So it's that whole kind of area. There's 1, 2, 3 atoms in the bond, basically. And if you didn't, I'm afraid there's no mark. Okay, B. Sunflower oil is used to make biodiesel, which contains methyl esters. The fatty acids in triglyceride molecule are reacted with methanol in a process called transesterification. After the reaction, 2 liquid products form which naturally separate from each other. The methyl esters float on top of a more dense liquid. Well, this is a really, really complicated way of asking you to, is basically asking you what is in a fat. Okay, this is super. They really tried to make this confusing for you. Okay, what it says is the fatty acids react, and then they become methyl esters, and then they float. Okay, so the fatty acids react, become methyl esters, and float. What else is there? Well, if we've taken out the fatty acids, yeah, and those have floated up, what is the other bit? It's the glycerol. Okay. Super weird way of asking you that, but yeah. That's one of those questions where you have to sort of stay calm. You haven't heard about transesterification and methyl esters, and you have to just read the sentences again and kind of think your way through it. Okay, C. Living organisms have many uses of triglycerides, one of which is the production of phospholipids. 3 other functions of triglycerides in living organisms. Well, there's loads. Okay, insulation is one. If you think about a seal or an aquatic mammal, or even humans, we use triglycerides for insulation under our skin. Another one is energy storage. And another one could be, let's think. Oh, okay. Protection of organs. Okay, a lot of organs have a fatty layer around. If you've dissected a heart, you see there's a fatty tissue around it that protects the heart, also around the kidneys. I think also we could have had waterproofing as well, but I'm not entirely sure about that because I don't know if that would count as inside an organism. It's on the feathers of a duck, for example. But is that inside or on? I don't know. Let's have a look. Energy storage, thermal insulation, electrical insulation. Okay, what this is talking about, this is actually talking about Schwann cells. So when I haven't done this, if you're a year 12 student, you haven't done Schwann cells yet, but when you do nerves in year 13, you will see that Schwann cells need to have use lipid to insulate the sort of flow of current, really, or flow of electricity, or the message, the electrical impulse in the nerve is what I'm trying to say. Okay, so those are the marks there. Pause if you need to have a double check. And then we'll move on. All right, okay. This question, notice I dropped in a picture over here on the right because I want to talk about that in a second. But the question here is about these 3 different methyl esters. Remember, we talked about these methyl esters that form from the fatty acids in this chemical reaction. Describe and explain the pattern of melting point of these 3 methyl esters. Well, the thing that is different about all of these, well, first of all, let's look at the carbons. It's the same. 19, 19, 19. The same. What's different then? It's not the oxygen that's the same, it's the hydrogens. 38, 36, 34. So this is all due to the number of double bonds, okay? So as we go down this way, I'll just bullet point them, we'll look at the mark scheme. The number of C double bonds increases. So therefore, they are becoming more unsaturated. And we can also say there's less hydrogen. So the molecules, this is all really the first bit. So the molecules are kinked. If we look here, these are the saturated fatty acids. They have a sort of straight structure. When we introduce a double bond, that's these highlighted bits here, we get a kink in the molecule. Okay, so instead of straight, it goes down and then it kinks to the side like that. That means that when you try and pack them together, they don't pack as tightly, which means that they have a lower melting point. Molecules are kinked, so they pack together less tightly, which leads to a lower melting point. Right, I almost didn't do this actually. I'm just gonna, like, it's always a good idea to sometimes look back at the question once you finished. Once you think you finished it, just to check, did I actually answer it? Didn't really talk about describe, did I? So I must say that as you move down, the melting point decreases, okay? It gets lower. Remember, this is a negative number, but it's a low negative number. Okay, let's check the mark scheme, make sure that we got the marks. Fewer hydrogens, more double bonds, less saturated gives lower melting point. And that's the sort of description. This is the D part. And then this is really the explanation part is down here. So describe and explain. More kinked molecule, less uniform packing together or packed together more irregularly, if that's a word, irregularly. Okay, moving on. Now the question is about the phospholipid bilayer. Allow the formation of membranes. Okay, so we need to talk about hydrophilic and hydrophobic. So we must, I just draw a quick section of it. There's just two of the fatty acids. We must talk about hydrophilic heads. We need to talk about them facing out. We need to mention the fact that there's water outside the membrane, so the heads face out into the aqueous environment. And we say that the hydrophobic tails face inwards or face towards each other to sort of to face away from the water to form a hydrophobic environment. So the mark scheme says fatty acid tails are hydrophobic, phospholipid bilayer formed, fatty acid tails on the inside pointing inwards. I've also seen the same question in previous papers. There's also given marks for saying that the hydrophilic heads are facing into an aqueous environment, but I think in those questions it might have been a 3 max or a 4 max, so there's more things you can say. I think on that one I would have got 2, but it is also sometimes worth saying the obvious. I think I forgot to say that it is a phospholipid bilayer in the membrane, which is key statement. All right, moving on. Okay, so for this one here, I actually found the original picture online because it's in color and a bit sort of higher resolution, so I thought I'd show you that. So this is the original picture here, taken from the internet, and this is where they've dropped it into the exam paper. So, okay, first of all, the first part of it is calculate the magnification of this image. All right, now I have not got a ruler, so this is a bit difficult, but the first step is to measure how long this is in millimeters. So in millimeters, that is, if we jump to the mark scheme, I'm sure it'll tell us, 21 millimeters. Okay, I'm going to jump back before we look at the mark scheme again. Okay, so this is 21 millimeters, but it says it's 60. Okay, so now we just remember that magnification equals the image divided by the actual. Okay, so the image size is 21 millimeters, but I'm going to convert that straight into micrometers because I want my calculation to be in micrometers. So 21 millimeters is 21,000 micrometers, and 60 micrometers is there. So 21,000, divided by 60 is 350. Tick, tick. Okay. The cell labeled T on figure 23 is a parenchyma cell. Parenchyma. which carries out photosynthesis and stores starch. It's just why cell T and the cells surrounding it can be classified as parenchyma tissue. Well, they are, I would say that there are many cells of similar structure and function grouped together. I hope that's enough. Or do I need to say that many identical cells? I mean, well, let's see. Yeah, fine. Similar cells working together with common, same, similar named function. Okay, so we need to both need to see both working together and same function. I don't think I said working together. Okay, so I don't get the mark. Must say for the definition of a tissue, working together. Okay. I need to remember my tissue definition better. Working together. All right, name the two tissues labeled Q and S. Well, let's actually just talk about these now for a minute. Okay, so always on the outside is the phloem, which is Q. And over here, that's these cells there. S is the xylem. That's these cells here. There's big empty xylem there. And later on, we're going to be asked to talk about what R is as well. There it is. So R is the, well, there's two ways you can call it. You can either call it the cambium, which is the sort of name of the layer, or you can say it's meristem tissue. So that's in the middle, as this layer here, okay? Yeah, cambium or meristem. So the way to remember it is just when you're going from the outside in on the vascular bundle, once you get into the vascular bundle, first there's this layer of kind of packing cells here. Oh, can I remember what it's called? I'm not even sure if I can. It might be called sclerenchyma. I don't think you need to know that. It's basically a sort of packing cells to kind of protect the vascular bundle as well, I guess. Then you have the phloem, then you have the cambium sits between the phloem and the xylem. The xylem is the most inner layer. Okay, so yeah, so Q, S, let's just look at the mark scheme. So yeah, Q, phloem, S, xylem, and C is either cambium or meristem or meristematic tissue. All right, moving on. 24, here's a DNA nucleotide. The circle component is, what is it? Is it a sugar? Is that good enough? Is it a pentose sugar? Is that good enough? Be more specific, it is deoxyribose because this shows a DNA nucleotide. So you know what type of nucleotide it is, so deoxyribose. And the type of bond labeled X, phosphoester. Phosphoester, I think. Am I going to get the mark though? Because if I don't get the mark, I'm going to be annoyed. Okay. It says phosphodiester. Let's just say phosphodiester. I disagree with this I disagree with this OCR. Shall I tell you why? Yes, I'm going to tell you why. The reason why is because technically a phosphodiester bond has to have two bonds. So the phosphodiester bond is actually the bond that links one deoxyribose through the phosphate to the next sugar. Okay, that's a phosphodiester. Technically, this is a phosphoester bond. OCR are wrong on this one, but they want you to say phosphodiester, so just say phosphodiester. Okay, so now we're on to this next bit. Similarities and differences between the DNA nucleotide and the molecule of ATP. Now I dropped in this picture of ATP here, slightly cut off here, but this should be adenine here. So similarities, they both contain adenine and they both contain phosphates. You could also say they both contain a pentose sugar. Now, if we want to drill down into that, the pentose sugar could be one of the differences actually, because here we've got deoxyribose in DNA versus ribose in ATP. So ATP has ribose. Also, ATP has 3 phosphate groups. I think that's the only other difference I can think of. So let's look at the mark scheme. Okay, so here we are. They both have a pentose sugar. They both have adenine. They both have a phosphoester bond. Yeah, all this stuff. Phosphates. ATP has 3 phosphates or 2 additional phosphates. Deoxyribose replaced by ribose. There's an OH group on carbon 2 of the sugar. Okay, so that's describing the difference between. This is the difference between deoxyribose and ribose. So on ribose, we have an OH group on carbon 2, but on deoxyribose, we don't have an OH group on carbon 2 of that pentose sugar. Okay, moving on. Right, Downy Birch. Here are some Downy Birch. Now, Downy Birch has sort of fine hairs on its leaves. This name is kind of funny in a way, because pubescens, that's basically, the person who named this tree was saying that a birch leaf looked a little bit like that first facial hair that men grow as they're going through puberty. So that is why this tree is named pubescens. It looks like a pubescent face, I guess. Weird. Anyway, these hairs are between 200 micrometers and 500 micrometers long. So that's 0.2 to 0.5 millimeters long. In response to different environmental conditions. State the pattern of variation shown by leaf hair density. Now this is a weird question. I think I know what it's getting at, but it's pretty weird. The pattern of variation, I would probably say type of variation. And I think it's trying to say continuous, okay? Because leaf hairs, you know, in terms of their density and how many hairs per square millimeter, you know, you could have 100 hairs, 101 hairs, 102 hairs. It is continuous. It's not going to be discontinuous for sure. Okay, so if we wanted to measure the leaf hair density, imagine if we had a leaf like this, okay? And we wanted to count look at the hairs on the underside of the leaf. So this is the underside of the leaf. Well, we would have to zoom in and we'd have to sort of magnify a tiny little space. We'd zoom in like this. We'd look under the microscope. So we would use a microscope. We would use an eyepiece graticule. To calibrate and work out the size of the field of view. Okay, so how big is the field of view here? Of field of view of the microscope. At a certain power, and then you'd count the hairs, and then you divide number of hairs divided by the area equals the density. And I think it's worth saying that you repeat this, you know, at least 5 times on a leaf. Repeat this multiple times. Take an average. Let's check the mark scheme. Okay, microscope, yep. graticule to calibrate or calculate the area of view. That's what I said. Count number of hairs. Divide by the area to get the density. Yeah, I said all these things. Okay, right. I think some statistics is coming up. Spearman's rank. Yeah, Spearman's rank. Okay. Right, now this is helpful. They've already done some of the ranking for us. So when you have Spearman's rank, you look at the data and you work out which is the highest point. So 13.7 is the furthest leaf from the river, and it's given a number 1. And then 11.9 is the second furthest, and it's given a distance of 2. So now we need to work out the ranks of all these. So scanning down, 34 is number 1. And then we've got 133. Any more 33s? No. So I'll just kind of cross these off. This is going to be number 2. Any more in the 30s? Yes, we do. We've got that one, which is number 3. Then we're looking for high 20s. High 20s, 27 is 4. Anything else in the 20s? 23 is 5. Then that's... So now we work out the ranks there. Now we do the difference in ranks. 4 versus 2. The difference is 2. Now it really doesn't matter whether you put a minus or plus here because you're going to square it and it's going to get rid of the sign. But that's just going to be 2 there. That's going to be 0. There's no difference there. 7 to 7, there's 0 there. 10 to 10, there's 0 there. 8 to 4, there's a difference of 4. 3 to 3, 0. 8 to 9, difference of 1. 6 to 6, 0. 9 to 2, there's a big difference there. That's 7. 5 to 5, that's 0. All right, so that's going to be 4, 0, 0, 0. Oh no, 16, 1, 49. Okay. All right, so now we use the formula down here. Okay, well that's worth 2 marks for that. Now we calculate this, so it's RS equals 1 minus 6 times the square of the sum of the differences squared. So the sum of the differences squared, so we need to do 6 times 4 plus 16 plus 49 over n. And n is the number of things there are. So we count them 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There's 10 rows of the table, so it's 10, and then 10 squared minus 1, so that's 100 minus 1, 99. So therefore, get the calculator out. 4 plus 16 plus 49 equals, that's 6 by 69. I'm just going to do the working sort of step by step. So if I go wrong, I can still get some marks. So 69 times 6 equals 414. 10 by 99, 990. So 414 divided by 990 equals 0.48. Now, there's one thing which I haven't done. Actually, my working is not very clear. 1 minus that. So I've got to take this away from 1. So what's 1 minus what I've got in my calculator here? So 1 minus 4.181 minus 0.4181 is 0. Doesn't tell me how many decimal places, so I'll probably keep it to 2. Well, no, maybe I'll go to 2. 0.582. Let's keep it to 3 decimal places. 0.582. Let's check the mark scheme. I feel like I've gone wrong somewhere, but I don't know quite where. Is this paper? Come on, guys. I mean, unless I'm wrong. 4 plus 16. 4 plus 16 plus 49. That's 69. There's another mistake in this paper. This is awful. I'm going to phone up OCR when they open up Monday and tell them they need to sort their marking out. Because that is equal to 69, not to 70. And in their mark scheme, they've totaled it up to 70. So that's yet another mistake in this paper. So, yeah, who knows how many thousand people got marked incorrectly across the country because of that. Well done. Right. D. The students concluded there is a positive correlation between distance of the tree from the river and mean leaf hair density. Yeah, there is a sort of small positive correlation. So just reasons why. This could be that the leaf hairs help reduce water loss. And this is because the hairs on the leaf, imagine it a bit like hairs on my hand, okay? They trap a small layer of air around the surface of the leaf, which means that there is less of a gradient in terms of the water vapor inside the leaf and outside the leaf, less of a steep gradient, which means that the rate of transpiration will be limited. And this is necessary as trees that are far from the river are further away from a water source. Okay, so that's the idea. Let's look at the mark scheme there. There's no need for me to write it out in full. Here it is. Further away from river, less water available, transpiration causes water loss, hairs trap water vapor or reduce transpiration, and they reduce the water potential gradient. So those are all the things that we said. Oh, we've gone up to the top for some reason. Scroll down. Okay. In this investigation, they randomly selected leaves from 10 birch trees at varying distances from the river. Okay, well this is a bit weird. Randomly selected, that's a bit strange. So maybe choose, I can never spell choose. Choose, that's not right, is it? Choose trees at regular intervals, I would say is a good idea. So maybe pick 3 trees that are within 5 meters, and then 3 trees that are between 10 and 15 meters, and then 3 trees that are between 20 and 25 meters. Some sort of systematic approach. So maybe use a systematic approach to picking the trees. And the other thing is about the trees. You know, make sure that trees should be similar. So the trees should be similar size. You know, if they picked one tree that is like, I don't know, that big, that's not fair, is it? And one tree that's massive. So the tree should be a similar size or similar age as well. Or maybe you could even say similar sort of health, because sometimes trees can have, you know, have diseases, for example. They can be infected with a virus or something. Mark scheme says, well, we'll check the mark scheme in a second after the last question, because we're on the last question here. Another group of students repeated this investigation and calculated an R value of 0.589, so pretty close. The critical value, RS, at the 5% level for 9 degrees of freedom is 0.6. So, okay, so what does that mean? It's just below, isn't it? So the calculated value is lower than the critical value. Which means we have to actually reject this hypothesis. We have to accept the null hypothesis and say that there actually isn't a difference. There isn't a statistical difference with this amount of data. No statistical difference. Now, in real life, if you were sort of wanted to just, you thought, oh, I was so close and I'm not sure if we have enough data, then maybe you go back and you look at more trees and you make those changes that we said up here, and then you could look at it again. But again, you'd have to do some statistics. So in this case, we reject the hypothesis, we accept the null hypothesis, there is no statistical difference. Let's double check the mark scheme there. Yeah, reject the hypothesis, there is no relationship. Okay, accept the null hypothesis. Their conclusion is incorrect. I don't think I said this. I think I've only got 1 mark. So the conclusion is incorrect. Up here, yeah, that's everything we said. Okay, so that's the final bit for that paper. I hope you found that useful. You can flick back through that, have a look at any areas that you found difficult, because of course they reuse questions like this. And I will be trying to speak to OCR about some of those mark scheme issues, so I can clarify those with you and see what they were trying to say, or why they were thinking that way when we come back to turn. All right, thanks very much. Bye bye.